Fields.
And also the people who are really quite really St. Compton's out there, they also find it very much jobs. Turns out top reading is pretty open.
Yeah, well, yeah, I mean, we just had that person for the here from Boston. Yeah.
Hello everyone. Welcome to our Friday seminar today, hosted by myself and the Streisfeld Lab. It's a pleasure to welcome Loren Rieseberg for today's seminar. Loren is a plant evolutionary biologist whose work has fundamentally shaped how we think about hybridization and speciation in natural populations. He grew up in Western Canada and earned his PhD in botany from Washington State University. After many years in the US, including at the Claremont Graduate School and Indiana University, he returned to Canada, where he is now a University Killam Professor at the University of British Columbia. His research combines population genomics with field and greenhouse experiments to understand how new plant species arise, how hybridization contributes to adaptation, and how these processes connect to challenges like crop improvement, invasive species, and conservation. His work has been recognized with many honors, including a MacArthur Fellowship and the Darwin Wallace Medal, one of the highest distinctions in evolutionary biology. He is also a member of several national academies, including the National Societies of London and Canada, the Norwegian Academy of Arts and Letters, American Academy of Arts and Sciences, and the US National Academy of Sciences. He's also served in major leadership roles across the field, such as the president of the American Genetics Association and the Botanical Society of America, as well as the director of UBC's Biodiversity Research Center, and currently the chief editor of Molecular Ecology. So please welcome me in joining Loren Rieseberg. Sorry, I dropped the microphone. First, I want to thank Gabriel for such a nice introduction, and also, of course, for all of you for coming. And Gabriel and Matt for inviting me and hosting me. It's been a real pleasure to visit. It's actually been quite a while since I've been to Eugene. Few of you may remember that Indiana University and University of Oregon have shared NSF IGER training program and in Evo. And so we had annual meetings that split between the two institutions. So sometimes they were in Indiana, sometimes in Oregon. And it was great because I got to know some really remarkable scientists here at Oregon. Some of them are still here. Bill Presto was just starting out at the time. Patrick Phillips was just starting out, but John Postlethwaite was there, was a real leader in that. And so it's a real pleasure to come back and see the people that I remember from those times. I also sort of feel a connection to Oregon because I use some of the software that people create here. As you'll see today, I use some Peter Roth software, which I found to be very helpful. And one of our associate editors, Angel, is here. You can see Angel, yeah. And then I'm sort of a selectionist, you'll notice, and so I've been really influenced by Andy Kern's work as well. So it's a real pleasure to be back and to see all these people that either I know or and I'm sure I've missed some people, but of course Matt Streisfeld has done a lot of influential work in evolution of plants. So it's a pleasure to be here. I'm sure I missed some names, but it's always good to be back. And today I'm going to be talking about a topic that I've worked on off and on and interested off and on for my entire career, and that is the role structural variation, really big, sort of the major structural variants, we used to call them chromosomal rearrangements, in plant adaptation and speciation. And I feel like in the last few years, with all of these genomes we now available, we're actually finally sinking our teeth into this in a way that really wasn't possible when I first started out. So I think, as all of you know, gene flow and recombination are often considered to be the largest obstacle to speciation in sexually reproducing species. And the reason for this is fairly simple, right? We know that forces such as divergent natural selection and genetic drift sort of push populations apart, and gene flow and recombination homogenize them. So these sort of opposing forces. And I've illustrated this here with this simple diagram where if a yellow flowered daisies, a population of yellow flowered daisies comes into contact with a population of red flowered daisies, of course, all things being equal, but in a few generations they will become homogenized. And this is a problem. This basic antagonism between natural selection and recombination has been known for more than a century by evolutionary biologists. In fact, the early critics of Darwin's theory of natural selection raised this as one of the main criticisms of Darwin's theories. And they used different language. Basically, they talked about how they're concerned that that traits that were favored by natural selection could not become established because of what they called genetic swamping or cross-mating with maladaptive individuals. But they really were talking about this basic antagonism that we continue to study today. And they came up with a solution. And it was a good one, right? And I think all of you know the solution. It was geographic isolation, right? If you have this yellow flowered population separated by some kind of a geographic barrier from a red flowered population, then gene flow and recombination are no longer possible, and so they're no longer a problem. And this was very enthusiastically received by many evolutionists. In fact, prominent evolutionary biologists in the 20th century, such as Ernst Mayr, suggested that, in fact, geographic isolation was a requirement for speciation. And many of these evolutionists, to the present day, well, not to the present day, but also sort of use fairly arrogant language in talking about Darwin's lapse, basically suggesting that Darwin didn't understand the importance of geographic isolation and speciation, and this was a major flaw in the origin of species. And so I was writing a review on speciation with gene flow last year. And I decided that I should go back and read some of Darwin's early papers. And I discovered a rather different picture. In fact, that I think Darwin's theory about geographic isolation was probably more nuanced and more sophisticated than many of his critics, ranging from, well, I'm not going to go through the critics. I don't like to criticize people too much. And so here's the funny thing is that Darwin's views actually changed over time, and they were influenced mainly by botanical observations. And so this is one of the reasons why I was particularly interested in it. So this is a quote from while he was, as you know, Darwin was preparing his book for many, many years. And in 1837 through 1838, he wrote that why are species not formed during ascent of mountain or approach of desert? Because the crossing of species less altered prevents the complete adaptation that would ensue. That is, if you have full geographic isolation. So he very much understood how geographic isolation was key to, and that there was this antagonism between selection and gene flow. And then he, as I think all of you know, Darwin was corresponded with hundreds of other scientists, thousands of letters, and he corresponded quite frequently with with botanists. And he learned in this correspondence, this was with people like Alphonse de Candolle and others, he learned that in fact, in many cases in plants, closely related taxa of different forms often were found geographically adjacent or even growing amongst each other and somehow maintaining distinctness. And so after, and he noticed the same thing with other less vagal organisms. And so in 1854, he writes, no doubt here comes in the question of how far isolation is necessary. I should have thought more necessary than the botanical facts seem to show. And then in his, of course, famous monograph, he writes, isolation by checking immigration and consequently competition will give time for any new variety to be slowly improved. So he very much understands the importance of isolation in adaptation and divergence. And then he writes, though, and he's very careful about this, may sometimes be of importance to the production of new species. And he also mentions that in plants and other less vagal organisms, isolation seems to be less important than in highly vagal animal species. And it turns out that Darwin was absolutely right, in my view. The genomic evidence that we've been accumulating shows that speciation with gene flow is actually seemingly quite common, supporting Darwin's views. And so what I'm going to talk about in this talk is, how does this happen? And there's an emerging solution. I should say this is not the only solution. There are other solutions as well, but this is the one I'm going to be focusing on, and that is structural variation. We now know that inversions and other chromosomal rearrangements suppress recombination in heterozygotes, and this allows or maintains sets of linked alleles. And these, of course, might be involved in reproductive isolation or in local adaptation. And this is, I'm using an illustration here from a paper by Mark Kirkpatrick, just showing what an inversion does. Here you have an inversion for two different traits, habitat adaptation and flowering time. And what happens is that in this particular inversion, there's recombination, but the recombinant gametes are all inviable because of duplications and deletions. You can get some small amounts of recombination through double recombinants, but it's quite rare. And then, of course, your viable gametes are the ones that are not recombinant. This particular model, though, is not necessarily always the case. There are many cases we're finding increasingly that inversions that are involved in local adaptation often show no crossovers at all. And I'll talk a little bit about why that is, because that's a puzzle I think that's really important to understanding how these rearrangements play a role in speciation. Because if they do actually produce these inviable gametes, the fitness effects can be quite large for a large inversion, up to 50%, which makes them very difficult to establish. So here are some key papers, and of course, I left out lots of them, and included one of my own, which is probably, self-aggrandizing. But anyways, these are some of the key papers I think that sort of influenced how we came to this conclusion. And I think in sort of discoveries or paradigm shifts, I'm not sure it calls it paradigm shift, but shifts in thinking about topics in evolutionary biology can take a long time. Sort of we accumulate evidence from many different sources before we begin to change our minds. And so I thought that this was, so when I started out as a grad student, we thought about chromosomal rearrangements as being important primarily because of their effects on fertility. That is, we noticed that when species were crossed that differed in translocations or large inversions, often there was a large loss in fertility. And so there are entire books written about promoting that these chromosomal rearrangements played a key role in speciation, mainly because they cause hybrids to have to be sterile. And but in the mid-90s, this idea began to shift a little bit. In the first paper, which has been largely ignored, that I think was important was a paper by Trickett and Butlin in 1994, and they ran a really simple simulation that in some ways I think was self-evident, but no one really had thought about it. And that is that if you had a locus under linked loci under one under divergent natural selection, this would be involved, for example, in local adaptation, and one causing assortative mating, that together these would, that an inversion that suppressed recombination between them would aid speciation. And that some ways seems self-evident, but at the time, no one was thinking about that. And I had been, I was influenced by that paper, in part because I had been looking at the effects of chromosomal rearrangements on gene flow between different sunflower species. And I was noticing that the rearrangements, this is both translocations and inversions, were having these inordinate effects on gene flow. You'd have part of the genome that had these rearrangements, there would be no gene flow at all, or very, very little, and then lots of gene flow across the species barrier where the genomes were collinear. And so I wrote a review suggesting that we really should be thinking about recombination suppression rather than the reduction in fitness, or at least in addition to the reduction in fitness. And in that same year, Mohammed Noor, who's a prof at Duke, wrote a really influential paper showing that how inversions could maintain hybrid incompatibilities or genic incompatibilities in the presence of gene flow. And this was important because if you have a typical Bateson-Dobzhansky molar incompatibility, in a hybrid zone, you're going to actually end up with favoring the ancestral state. So there's no, the incompatibilities are washed away by gene flow. And so this provided another sort of important component. And I remember when I wrote this paper for a speciation special issue, one of the reviewers said, well, how did these rearrangements become established? I didn't know. I wrote a small paragraph about co-adaptive gene complexes, building on some of Brian Charlesworth's inversion papers. But the real breakthrough came in this paper by Mark Kirkpatrick and Nick Barton in 2006, where they showed that suppression of recombination between locally adapted alleles actually favors the establishment of inversions. And this was a breakthrough because it really began to explain how all of these inversions that we were beginning to see with genomic data, how many of them were becoming established. And there are other important papers, but this is, I think, some of the influential early history. So what I'm going to do is talk about how these ideas sort of formed and then became sort of taking empirical data that sort of supported or helped or illustrated that in fact this process really is happening in healthy speciation with gene flow in sunflowers. And so I'm first going to talk about a large-scale study we ran several years ago looking at the genetic basis of adaptation in sunflowers. And then I'm going to go through two case studies of speciation with gene flow, one at the very early stages with the silverleaf sunflower, and then one where speciation is almost complete, involving two dune and non-dune ecotypes of the prairie sunflower. I'm then going to talk a little bit about the divergence level of chromosomal inversions between the different haplotypes, and suggesting that maybe this is the key to the lack of recombination and how they arise. So I'm going to argue it's because they arise through introgression, another component of my research program. I'm also going to talk about, so recombination is usually considered to be something that's good, right? Because it helps to bring beneficial alleles together and deleterious alleles together, increasing the efficiency of adaptation. So how do sunflowers avoid the maladaptive effects of inversions? And then I'm going to end by looking at the distribution of inversions across the tree of life. We recently assembled more than 1,000 genomes of plants, animals, and fungi, and we're beginning to get an idea of that maybe there's some differences between kingdoms in how these things operate. So just a quick introduction to sunflowers. Wild sunflowers, there's about 50 species across North America. The populations tend to be very tightly adapted to their local environments. And this is important to speciation because in plants, local adaptation is often the first step in speciation. And I want to give a shout out to one of my former postdocs, Dylan Burge, who's a remarkable botanist. He had a red truck that he drove across North America, collecting sunflowers. And he also didn't just collect the sunflowers, he dug up the soil. He got 4 soil samples of each site so that we really knew we could characterize the soil that these were found in. And there were sort of 2 widespread sunflowers, the common sunflower and the prairie sunflower that are parapathic or sympathic across most of the central and western US. And then the silverleaf sunflower is the narrow endemic to the coastal plain of Texas. And so this just gives some idea. This was this initial large-scale genetics of adaptation study. We grew about 1,500 plants in common gardens in UBC. We phenotyped them for about 80 plus phenotypic traits. We analyzed the soil for about 15 characteristics, and we also downloaded a bunch of climate variables from the World Wide Web, and then we sequenced them and called a bunch of variants. So that's the data set we'll be working with. These are the 3 main postdocs at the time who did the work, all very talented. Marco is an assistant professor at UBC now, so I'm glad not to lose him entirely. Greg is a assistant professor, well, now associate professor at the University of Victoria. And Natalia is now running the molecular biology lab at in the Biodiversity Research Center. So I'm very pleased to sort of kept them in some ways close by because they're all super talented. So we did 3 kinds of analyses, and I'll introduce them briefly. I think you know most of you are familiar with these, but one kind of analyses are these genome-wide association studies, and here you're just looking for associations between genetic variants and phenotypic variation. And so this is an example where the trait is these ultraviolet nectar guides or these UV bullseyes. You see, this is what we see is this pretty yellow flower, and then the insects see these bullseyes. And so this is a GWAS for Helianthus annuus and the common sunflower. And so just so that you do understand these things, each of these is just a different chromosome. And then the significance threshold levels are shown here. This red line is the von Ferroni corrected significance threshold. You'll see a lot of these. And then here, these are just snips at the bottom, and you can see how there's one giant hit, and this maps to a homolog of Arabidopsis gene middle-11, which is involved in the regulation of flavanol production in Arabidopsis, and of course flavanols absorb UV. And Marco did a bunch of functional work showing that indeed this was a cis-regulatory mutation that is responsible for most of the variation in UV patterns. But you can see how this works. This is really what you would like to see. Most of the time you don't get such a nice pattern. And then we also did something called environmental GWAS. Sometimes it's called genome environment analysis. This is this GA. And here you're looking for, it's the same kind of Manhattan plot where you have your chromosomes, a significance threshold. In this case, you're looking for associations between features of the environment and genetic variants. And so the rationale behind these is that the loci that show these strong hits are involved in adaptation to these different environmental features of the environment. So this is a case where you see a strong hit to an Arabidopsis gene called HIT1, which is heat intolerance 1. And so we presume it's involved, like in Arabidopsis, to high adaptation to high temperatures. And one of the things that we found, I think I had my slides not quite in the right order. Sorry, I made some changes last night. But so one of the things we found is sometimes we actually would see large signal plateaus. This is an example here in the prairie sunflower Helianthus pilaris to cation exchange capacity, which is a measure of soil fertility. And so what you see is these large signal plateaus. Those are actually bigger than they seem. For example, the largest one there is about 60 to 7, chromosome 14 is about 60 megabases, so it's a fairly large region. The largest one though is here on chromosome 13. It's about 101 megabase. To give you some perspective, that's about 3/4 the size of the Rhabdopsis genome. It's about half the size of the minerals genome. I guess not quite half, but maybe yeah. So it's quite a large region. The other approach we take, I was actually going to put this on earlier because I didn't have it in my talk originally, but it is something we did a lot. And it uses Peter Ralph's program, which we found very effective in sunflowers. But this is, we also use this low struct program where you're looking for variation in the genome in local population structure. And it provides a really powerful way for finding regions of the genome, large regions of the genome or windows that are under selection. That are showing patterns of local adaptation. And this works wonderfully in sunflower. This is an example here from chromosome 5. What you're looking at here is you're looking at the multi-dimensional scaling plot, MDS. Yeah, anyways, so you're looking across the genome here, and you're looking for each of these windows, you calculate the PCA or population structure. And so you can see all the windows basically show the same population structure until you get to about 150 megabases on the chromosome. Then all of a sudden, outbreaks is a very different population structure. And these are mostly due to large structural variants. And so we use this approach as well. And when we actually, now we have genome sequence for almost all of the species are involved here. And what we find is that most of these regions that we detected in our GWAS or low struct turn out to be inversions. This is that inversion we just showed you on chromosome 5, that apple block in chromosome 5. This is that large 101 megabase apple block. You can see it's a large inversion. There's also another inversion on the same chromosome that we see sometimes. And when these are together, they often seal off the entire chromosome for recombination. So we made Marco made near-isogenic lines for this apple block, and it turns out that the near-isogenic lines are just for the entire chromosome. We can't seem to break up the two inversions. And we find that these haploblocks underlie traits associated with local adaptation. To make it simpler to see, we divided these up into sort of different categories of phenotypes: plant architecture, carbon nitrogen content, development, inflorescence traits, leaf traits, seed traits, and then climate traits and soil. And so most of these are actually involved in local adaptation. And even ones that don't show up here, when we do more fine-grained analysis, we find that population structure in some ways has prevented us seeing, we see strong associations, but they're not significant because of population structure. So if we use different approaches, we do find a hit. So now I want to go through this first case study. So this involves the silverleaf sunflower. There are two ecotypes in the silverleaf sunflower that were discovered by one of my former grad students, Brooke Myers. And she found them when doing a common garden at UBC, where there was this tall, late flowering sunflower that actually would start flowering just about the time it started to snow in Vancouver, very late. And then this other one, short, early flowering one that would flower in July. And this one turned out to be only found on the barrier islands of the Gulf of Mexico, not Gulf of America. And the other tall, late flowering one was found on the coastal plain, a little bit inland. And not surprisingly, she actually did some selection tests and showed that these were under divergent selection. It's a much milder climate here on the Texas coast. So these things, these tall ones, basically shut down growth during the middle of the summer when it's super hot. And these ones just flower as soon as they can and flower for the entire season. And when we did the genetics, the GWAS, we found that there was this enormous 30 megabase region that controls flowering time. It actually is responsible for about an 80-day shift in flowering. And there's an interesting thing is you find that, notice this gap in the GWAS signal, we'll get to that in a bit. And so if you look at this more closely, we found that there was actually a 10 megabase region where there was no recombination at all. And this region contains an important gene family. This is the FT gene family, or flowering locust T. And this is the protein of flowering locust T is the floragen hormone that moves from the leaf to the apical meristem to trigger flowering. And this particular gene, FT1, is a gene that Ben Blackman, one of my former grad students, cloned, and it was the first sort of domestication gene in cloned sunflower, and it results in later flowering because of friendship mutation. And so we're really interested that it turns out that this FT1 gene turns out to be missing in the late flowering form of Helium sargophyllus. So same gene as in the domesticated sunflower, and seemingly a loss of function mutation. And so Marco did all of the functional work, and he showed that indeed the late flowering FT1 would not amplify in genomic DNA, also nor from cDNA, and he also transformed or complemented the Arabidopsis mutant and found that indeed the early flowering allele was functional. And so we are fairly confident that this is the gene that's causing the shift in flowering time. So what does this mean in terms of reproductive isolation? So we initially thought with this very large difference in flowering time, it would have a huge impact on reproductive isolation. This is sort of the first step in speciation in this group. And so one of my former grad students, Mariana Pascal, went down to Texas and measured the effects of this flowering shift on reproductive isolation, actually genotyping them for this FT mutation. And sadly, it has a very minor effect on reproductive isolation, something I really hadn't thought through. So it is true you get very strong reproductive isolation on the coast. This is also inland, but we measured this in populations that were polymorphic for both haplotypes. We found that the reproductive isolation was only strong in November. That's when the seed were produced, so the isolation itself would have been in September. And then on the island, where they flower very early, the only strong isolation was in July. And of course, this makes sense because you get the overlap in isolation and flowering for the rest of the season. So this is actually a very weak reproductive barrier. That surprised us. So we looked at genetic differentiation between the inland and island forms to see if there was much going on. And this is a measure of FST across the genome. So FST is a common measure of genetic differentiation. And what you see is that we do find that there's not that much differentiation. But the differentiation we do see is this is the chromosome 6 FT, that big haploblock I showed you. Here's also another inversion of parent chromosome 10. You find that much of the differentiation we do see is in these haploblocks. And so I think what we're seeing here is just sort of the very earliest stages of speciation, whether this ever goes on to form full-fledged species. Speciation of the gene pool is post complete. And this is with these non-dune ecotypes in Colorado. And these ecotypes were discovered by one of my former grad students, Loren, who's now a prof at University of Colorado. And what he found was they had these sunflowers on the dune, and they had these enormous seeds, whereas the ones off the dune, which we created called dune non-dune ecotypes, actually had quite small seeds. And so this is the Great Sand Dune National Park, and these are the largest dunes in the US. And so here you're seeing the dune is here. This is the sand sheet where you see the typical what we call non-dune ecotype, and then the dune ecotype is there, and then you have the forest behind, or the mountains behind them. And there, in addition to seed size, the plants flower later. There's also greater nitrogen use efficiency, so dunes are not water limited, but they are very much nutrient limited. So that's probably that plus disturbance are the two strongest selective mechanisms, selective pressures, and the larger seeds allow dune sunflowers to keep the dune seeds from being completely buried by sand. The larger seeds go to the top of the surface, but it also gives them the nutrient reserves to tap into water and nutrients lower in the sand dune. At least that's the adaptive story. And so one of my postdocs, former postdoc, Rose Andrew, she using rad-seq data that was generated by a company developed here at Will Prestos, I think. And but anyway, she measured both the fused coalescence models to look at divergence times. These ecotypes seem to have diverged very recently. And she also showed that there were modest levels of gene flow. So this is speciation with gene flow. There was equally these models. And slightly more gene flow from the dune to the non-dune, which makes sense because dune seeds actually survive in non-dune habitats, but you get no survivorship of non-dune seeds in the dune habitat. So the only gene flow is through pollen for that direction. But anyways, gene flow is moderate, but high enough to prevent differentiation through drift, but not high enough to prevent differentiation through selection, as we'll show in a minute. And so Kate is the author that carried out some reciprocal transplant experiments. I should say, I think I've ruined a number of people's careers on these sand dunes because this is a terribly difficult habitat to work in. Most experiments failed, and there were two reasons for this. One is the obvious one. Your experiment gets covered by sand, if you're unlucky. But the other one is, these are often the only green things out there, and so herbivories, it's just devastating on the dunes. It's remarkable how many organisms are out there. And then also here's the sand dune, but she carried this out successfully. And so what she did was she measured the strength of reproductive barriers across the life history. And what you see is that they actually get very strong overall isolation between 0.993 and 0.996. So these are almost completely isolated. And the biggest barrier, turns out, is immigrant inviolability, is because the dune genotypes don't do well on non-dune soils and vice versa. There was a small difference in flowering time, but as I mentioned, flowering time is not a great reproductive barrier. A bigger barrier was pollinator isolation. That's because there are very different pollinator communities on the dune and off the dune. And so in some ways, it's almost a consequence of the difference of immigrant inviolability. We saw some evidence of conspecific pollen precedents. What's remarkable here is that dune moms can recognize dune bad pollen and prefer it. And I don't know how they do it, we still haven't figured that out. And non-dune moms also prefer non-dune pollen, but not as strongly. And we didn't see any difference in sort of class of hybrid inviolability or hybrid fertility. We did see a strong selection against hybrids. And so how does this relate to reproductive isolation? Well, all of the key traits that differentiate the dune ecotype are found in these haploblocks, which are inversions. And this includes seed size, flowering time, also soil adaptation to low fertility soils. We also, because Kate had planted out all these hybrids, we also could measure the direct fitness of the inversions themselves. And so we found that in most cases, the inversion heterozygotes were selected against, which is really important for that model of Kirkpatrick and Barton for the establishment of inversions. And then finally, if you look at FST across the genome, you notice how different this is from the Argyrophilus example. Now, we see very high FST in certain parts of the genome. Almost all of these are these inversions. And so it seems clear that it is the haploblocks that are defining these ecotypes. So you might ask, well, how does speciation occur so quickly? Well, we think what happens is that these haploblocks were recruited from standard variation. For example, in the Great Sand Dunes, just during the Wisconsin glaciation, it would have been too cold for any sunflowers to survive, right? So they must have collapsed down into the non-dune area, probably went further south, and then they came back and colonized the dunes using the same variation over and over again, very similar to the stickleback story. And we suspect that this has been going on for maybe multiple millions of years, where each time the dune ecotite forms, it's probably maybe a little more strongly isolated than it was the last time. So one of the interesting aspects that has fascinated me is that all of these haploblocks tend to be very highly divergent from the two forms of the two different haplotypes of the inversion or other structural variant. And this is an example here of that haploblock and from that contains the FTG, the 5, and you can see here that the early article haplotype, this is the functional. This contains the functional gene of FT, was actually derived through regression from Helianthus annulus. Fairly recently, we think probably in the last 50,000 or so years, whereas this is the haplotype found in the late flowering articles, which clusters already expect in terms of phylogeny. And we think this is what explains all of them. And so why does origin through introgression matter? It took me for a long time to figure this out. So this is just showing that this figure, you don't have to spend too much time on it, because it's just showing that most of these, that the inversions in the dune and non-dune ecotypes, none of them seem to have any effect on fertility. And so I think what's going on is that if you have highly divergent haplotypes, you don't get synapses during meiosis. That is, no inversion loops forms, there's no recombination. So no fitness loss due to the production of a viable pollen caused by recombination. And we think that's the key to their establishment. Even though you have the Kirkpatrick and Barton model suggesting that inversions are favored due to the suppression of recombination, there's no way it can explain a loss of very stringent conditions to explain a loss of 50% fertility. It just doesn't happen. Talking a lot with Mark about this. I think the key is that when you drive these things from introgression, you already have very divergent haplotypes. And so you get complete suppression of recombination without a loss of fertility. I think that's key to establishing these things. There's an interesting paper by Jeff Fader back in 2011 that I thought was interesting, because Jeff had noticed that inversion in Rieseberg, this is the apple microbiomes that are classic symptomatic speciation stories, but the inversions that were involved in this turn out to have been derived through introgression. He called them derived through or something, but anyways. And so his argument was, and he created a model showing this, is that geographic isolation prior to introgression actually facilitates the sort of buildup of sets of locally adaptive alleles. And I think that's the case. However, I think what's more important is actually the divergence of the haplotypes that probably eliminates synapses during meiosis. Okay, I might have to move a little bit more quickly since we're running a bit late here. So how do sunflowers, I do want to address a couple of these things. So how do sunflowers avoid the maladaptive effects of recombination suppression? And I'm showing this why recombination is beneficial, right? If you have a chromosome with both a deleterious and a beneficial allele, it's very hard for selection to act on those because you have those opposing effects. Recombination can bring deleterious mutations together, which becomes easier for selection to eliminate them, and also, of course, allows selection to favor the beneficial mutations when they're brought together. So what's going on in sunflowers? We shouldn't have all these inversions because it should be bad. And what we find, though, is when we actually, I'm going to go through this really quickly. What we find, though, is that when you look at the accumulation of deleterious mutations, in this case, we're looking at the accumulation of loss of function on mutation. These are nonsense mutations. You can see that there's actually no difference between the inversion deleterious mutations and the inversions relative to the rest of the genome. And we had to classify these in terms of the recombination rate in those genomic regions. And so what's going on? Why aren't they accumulating deleterious mutations like they should? And we think it's because inversions, remember, they only suppress recombination of heterozygotes, which makes them kind of special. Other structural variants are mostly the same, like deletions. Translocations, it's a more complicated story. And when you have inversions involved in local adaptation, at least in plants, the different haplotypes tend to be geographically separated. So they're mostly homozygote, not homozygous. And you can see this here. This U score is simply the measure of homozygosity. And you can see that inversions are much more homozygous than the rest of the genome, which probably explains why they don't build up deleterious mutations, because most of the time they're homozygous and they can actually eliminate the deleterious mutations just like the rest of the genome. So I want to end by looking at the distribution of inversions across the tree of life. Well, the eukaryotic tree of life. We haven't done this for microorganisms. And so one of my postdocs just went to public databases, mainly the Darwin Tree of Life project, but also GeneBank and so forth. And he pulled out all genomes that he could find that had both long read data and high C data with scaffolding. And then he assembled all of these genomes, did haplotype resolved assemblies. Each diploid, right, has 2 genomes, right? And then he compared the different haplotypes within each individual, because each individual are wild genotypes, so they're heterozygous. So you can get an estimate of lots of different genomic features just by comparing the haplotypes found within heterozygous genotypes. And so he just did one per species, right? And so what he found was kind of interesting. This is a big surprise. It turns out that inversions are more common in animals than plants, which was the opposite of what I had expected. But the inversions in plants are bigger. And these big ones that are involved in localization. I don't have time to spend very much long on this, but it also turns out that unlike animals, in plants, the long inversions actually are more divergent. They become more divergent, whereas in animals, the long inversions are haplotypes are less divergent. So a very different sort of evolutionary strategy. And then lastly, we found that plant inversions have fewer genes, but more transposable elements, and this becomes more and more pronounced the longer the inversion. And so the reason I think that this is important is that the regions of the genome that diverge quickly are going to be the regions that are rich in transposable elements or centromere elements and so forth. So I think that these probably are more likely to result in asynapsis during meiosis. That is, they won't recombine as much as in animals. So I'm going to conclude now. And I'm sorry we're running slightly late. So obviously, what I've tried to show is that sunflower ecotypes are differentiated by these massive haploblocks due to structural variants, mostly inversions. These contribute importantly to local adaptation and to reproductive isolation. They're mostly caused by these large inversions, and these act as recombination suppressors. And these are ideal recombination modifiers or recombination suppressors because they only suppress recombination in heterozygotes. And so this helps to avoid some of the ills due to the accumulation of deleterious mutations. And they play a major role in evolution by reducing recombination between locally adaptable alleles, as well as those involved in assortative mating or other forms of reproductive isolation. It's important to note that this mechanism actually works across eukaryotes because inversions are common in other groups of animals, plants, and fungi. One of the fascinating things, though, is they're actually more large and more divergent in plants, possibly suggesting a greater role in plant adaptation, which is sort of what Darwin suggested, for potentially a mechanism for those early observations that geographic isolation doesn't seem to matter, plant speciation. Of course, it could be that because local adaptation is more important, plants up where we see all of these inversions, there's some circularity in those arguments. And so I want to end by saying this offers what I think is a general solution for how speciation occurs in the presence of gene flow and recombination. And I don't want to say it's the only one, but I think offers a fairly general solution, and I think actually is very close to providing a mechanism for Darwin's observations. And I want to end by thanking all the people who did the work. This was a big team effort, but these are the ones who really played a large role. Barco, Natalia, Greg, Kate, Shu, and Peggy. And we do a lot of our work with the Tedesco Lab. And so here's a photo of our sprint group. And so thank you very much. I don't know if there's time for questions.
Oh, we got time.
Approaching the hour, but if you have any questions, please. You should feel free to leave. Yes.
So, Laura, you were talking mostly about diploid speciation, right?
Yeah.
So would you say that there's probably similar mechanisms besides diploidization in polyploids?
Yeah, so in polyploids, it's an interesting thing because in polyploids, you're right that the accumulation of many of the reproductive barriers is simply a consequence of the loss of duplicate genes. And Matt Hahn had a really interesting paper in 2016 suggesting that the divergent evolution of duplicate genes might not be fast enough to drive plant speciation. It's certainly a very interesting, clearly these inversions do accumulate in polyploids as well, but whether there's dominant a form of plays dominant role, I really don't know at this point. My sense is that, there's this argument that I've been involved in to some extent, whether polyploidy increases diversification rates or not. And there are two ways of looking at it. One is there's been this sort of idea that it's only after about 5 or 10 million years where what you find is that among very closely related things within a genus, you see that polyploidy is associated with a reduction in diversification rates, whereas above the generic level, it's associated with an increase in diversification rates. So there's this idea that you have to have this diploidization before you get that advantage of polyploidy. But the other thing that might be going on is that the loss of the lower diversification rate at the generic level is just because autopolyploids are included in the estimate, whereas we know that all the whole genome duplication, apparently almost all of them seem to be allopolyploids. It could be that if we just got rid of all the autopolyploids, that we would, that the two rate estimates would be significant. But that doesn't really address your question. Yeah, I'm assuming that these things do play a role in polyploid speciation, but there are other mechanisms that also are quite important in polyploids, and so I would generally think that but the other mechanisms are sensitive to gene flow. So like the divergent resolution of duplicate genes requires allopatry or self-immunity to that all of the genes, all of the examples of hybrid incompatibility is due to the divergent resolution of genes. I think all of them but one happen in sulfurs. It shows how gene flow can knock up that mechanism. I know that I think they'll help develop the theory and so on, but Mike Lynch, and I know it was that Garland Force, yeah, so I'll review.
So you mentioned this really interesting thing that a lot of these inversions that you're observing generally occur as homozygous.
Yeah.
There's a really interesting piece of literature from the late 70s and 80s in Drosophila, a series of papers by Stryker, who showed that some of the cosmopolitan inversions in Drosophila really showed overdominant.
Yes, I did.
is there any evidence of dominance when you look for heterosis?
So it's a really great question because, that, so in Drosophila, most inversions are polymorphic within populations and by the way, in sunflowers, when we look at populations where inversion haplotypes are polymorphic, we do see the increase in heterosis. But most in Drosophila, most of them all are, same thing in Heliconius where you see the overdominance. In sunflower, we don't have a lot of overdominance, but there is one intriguing thing that I didn't have time to talk about, but in chromosome 14, in that comparison of the fitness of heterozygotes, chromosome 14, the version on chromosome 14 actually had the heterozygotes had a positive fitness effect. So that's the one I look at. And that could be due to heterosis. But yeah, it only really is an advantage if you have for inversions that are, and this can lead to long-term balancing selection in populations. And so I think it's more going to be more of an animal phenomenon than in plants, where most of the inversions are strongly locally adapted and you have habitat segregation.
So you highlighted mostly kind of the larger, some really big versions, right? Many megabases. Wondering though, like in the sticklebacks, when we find, 2 or 3 megabase versions of 1, 11, 20. But then we have a lot of these really complex sort of nested in used for combination, but are really complex. So I'm just wondering whether you see those as well.
Yes, we do. I wrote a review paper a few years ago trying to make a big deal of the stepwise accumulation of these inversions within. You see these complex inversions, and some of those, like that one chromosome 5, that's actually 2 inversions back to back, and then in some cases it's a 3rd small one. So this is quite common to see. And that haploblock in Arbophilus on chromosome 6, it's a deletion plus an inversion plus other crap. Pericentric region really, due to an introgression. And so we had initially thought that maybe you'd see the stepwise, you see the evolutionary strata like you see in sex chromosome inversion accumulation, at least in some part we don't. There's no evidence of sort of the different time series. I still think that must happen. It happens in ants, where you see again the stepwise inversions, in some of the ant supergenes, like sex chromosomes, but we don't see it in some parts. But I think maybe still vaccine, I don't know.
Stay Loren.